Phil and Karen and Republican Liberty Caucus. I appreciate the opportunity to be here. I appreciate all of you for coming. We have a situation that should be of great concern to everyone, even greater concern to anyone that's a parent or a grandparent or a great-grandparent, if you're looking at the well-being of your posterity. The mural that you see on the slide here is painted on the side of a cafe in Los Angeles. I have a grandson that's a surgeon there, and he said, "Grandpa, when you come down to visit me, we'll go out for dinner." And so told him we'd do that. But it depicts a lot of the concerns that are expressed when you discuss genetic engineering. And so I'll cover some of those. There was a funeral in Mississippi a couple of years ago. Donnie knew that he had terminal cancer, and that this was around the corner for him, and so in his will he wrote out very specific instructions because he wanted to be remembered for who he was and not just somebody stretched out in a box sitting in front of the audience. And so he specified everything that would proceed in that funeral. So he wanted to be remembered relaxed, enjoying football. He had the TV set over here with playing or showing one of his favorite Pittsburgh Steelers football games. He had his Steelers blanket there, can of Budweiser and his Newport cigarettes, and he was just relaxed, enjoying the occasion. Let it become known, rumored around town, that he had allocated $30,000 in his will to make sure that it was just what he wanted in that process. And so after the funeral, as people were leaving, his wife Jolene was walking out and had her friend Jill came up to her and said, "Well, Jill, Jolene, I thought it was really an excellent funeral service." And Jolene said, "Well, it was just exactly the way he wanted it." Said he wanted to be remembered and relaxed as a friend, as enjoying life and sharing that. And Jolene said, "Yes, I agree that everything went very well, and I think Donnie would be very happy with the proceedings." And then Jill said, "Well, you know, I was just curious. There were rumors going around town, and I was curious how much this cost." And so Jill said, "Well, I had the expense for the minister to conduct the services, and I had the expense for the church, and of course the setup, there was some expense involved there." And she gave off the amounts that all of that had cost for the funeral home and that, and the bottom residual was $20,000. And so Jill said, "Well, you know, what was the $20,000 for?" And Jolene held out her finger, and she said, "For the memorial stone." And Jill said, "Well, what size memorial stone did you get?" She held out her finger and said, 2 carat. So we have more than a 2 carat problem when it comes to genetic engineering. Genetic engineering has been promoted as the solution to all of our problems. Doesn't matter whether it's global climate change, whether it's growing world population, or excess pesticides, or anything you want to come up with. genetic engineering is the solution for it. And you can go down that long list, and I have a few of the items up here. And now that we've been using genetically engineered crops for almost 20 years, you can go down that list, and every one of them are a failed promise. There isn't a single one of them that has increased yield, increased quality, or reduced pesticide use. It's been just the opposite. So that we have failed promises because agriculture is the management of an ecology, not just taking silver bullets. If you read the ads in the magazines with genetic engineering, they'll have all these silver bullets there that they're going to solve all these promises. And so agriculture, though, is the management of an ecology. It's not just shooting silver bullets at every little item that comes up. And you can't do one thing in this ecology without interacting and changing the interaction of all of those other factors. So you have the plant at the top, you have the physical environment on the one point, you have a very dynamic biological environment. If you step on an acre of soil, you're stepping on about 10 ton of living material. It's not just sand and silt and clay, minerals. It's actually a very dynamic system that is managed in this agricultural program. And of course then you have your pests and diseases and those things. So as we manage the ecology for agriculture, we're providing everything to support the plant and making the conditions as least conducive for the pests and diseases so that they're not a problem for us. It's that ecology that we're managing rather than just shooting silver bullets at every problem that comes along. With genetic engineering, it's based on fossil science. We threw out that, or we changed our concept of genetics 50 years ago. Genetic engineering didn't change. Genetic engineering is more like a virus infection than it is a breeding program. With the breeding program, you substitute entire groups of genetic material so that you have the regulatory and the promoters and the active aspects of that all functioning together. Genetic engineering, you're disrupting that and you're just splicing in materials. You're inserting them to disrupt the integrity of that genetic code, just like a virus does, or like the plant tumor bacterium does that's used to genetically engineer the plants, that causes crown gall, the agrobacterium bacterium, that you can attach genetic material to and it'll insert it in the cell and get the expression of that new genetic material. But it's no different really than a virus infection. And we can use that with the tulips. If you see the variegation in tulips, the different colors in tulips, many of those are actually a virus infection. When we cure the virus infection, you just have a solid color. And so you can get expression of genetic material, but it's very distinct and different than a normal breeding or hybridization program. In order to get that, we even have to have virus promoter genes. In other words, you can have genetic material, but if you don't have that part of the virus that's responsible for virulence, for expression of those genetic traits, you don't have any expression. So again, the concept of genetic engineering is based on the old philosophy, the old theory of genetics, that you have one gene for every function. And so when we started sequencing the human genome, we had anticipated 100,000 genes. Scientific American came out after the sequencing and said, where are all the genes? We have just a few more genes than an earthworm, just a little more than 18,000. That's because our concept and understanding of genetics 50 or 60 years ago didn't match the actual functioning of genetics as we understand it today. So that again, genetic engineering is more like a virus infection or a pathogenesis relationship than a normal breeding program which has all of the regulatory controls with it. It's a disruptive process so that you always have a yield reduction and generally a quality reduction. I don't know of any single genetically engineered plant that yields as much as the isogenic parent. Our yields have gone up not because of genetic engineering, but because of traditional breeding. And then those materials have been converted into the genetically engineered plants to express the trait. But there's always a yield reduction or a yield drag. And we used to talk about it all the time. It's been forgotten the last 15 years. The genes also, since they're just inserted, are not stable in the system. They're very promiscuous. They can be picked up by microorganisms, both in the soil or the environment as that residue decomposes, or in your GI tract when that food is consumed. That means then that you can start to produce some of those same entities or some of those same chemicals that the plant's producing. And it's been estimated that 95% of the population now will actually be expressing some of the Bt characteristics or toxins that are genetically engineered into many of our plants now for insect resistance. That's the Cry1A toxin. So that our current concept of genetics is that it's a spatial relationship of each genetic material, piece of genetic material, in relationship to its adjacent material as it's influenced by the environment. So as the environment changes, you have a different expression. One gene then may actually regulate or promote many different functions. That's why we have over 100,000 functions that we know of, but we only have a few more than 18,000 genes. Because as the environment changes, you have those other functions produced. If it wasn't that way, we'd be all eyes or ears or toes or just a blob of tissue, because as the environment changes during development, you have differentiation. But you have the same genes. It's just that the expression then is changed. In genetic engineering, you can see that there's a splicing where that red star is. You've disrupted the integrity of the genetic code, and rather than just one function then, because it's a spatial relationship as it's influenced by the environment, you have many functions that are changed. If you just look at the Bt, the insecticide genetic engineering, this is a toxin that's produced by soil-borne bacterium, Bacillus thuringiensis. You can look at the event with the MON18 Bt production, and you can see each of those colored items up there is a codon change. It's not just one. You've changed literally hundreds of of entities in that genetic component. You look at the BT11. It's not the same, but it's still the BT toxin. That's because we have no way of directing that material into the chromosome. It goes in at random. And so every time it goes into a different location in that helix, you're going to have different functions expressed and different functions changed. It's not going to be the same. We don't know how to get a duplicate of any single genetically engineered event. There are many changes that take place, not just the one that you may be targeting or looking for. So that in understanding genetic engineering, there are two factors that have to be considered. The first is just the intended or the unintended consequences and what their safety and effect on the plant is. This may be the consequences of where the insertion goes, whether the promoters are fully active, those virus promoters that are always associated and attached to the trait that is